Anything produced by evolution is bound to be a bit of a mess. After you hear today's lecture, you'll understand why that is, because evolution is a pretty random collection of mutation and selection. I think this lecture is one of the more exciting ones in the course because we now have tools that we can use to study evolution like we never did before. In the past, we just looked at things and tried to deduce relationships and what kinds of selections led to their emergence. But now we can sequence genomes, not only of viruses, but of hosts, and we can look at the interplay that goes on. And the second quote, around here, it takes all the running you can do just to stay in the same place. That's a reflection of these arms races between viruses and hosts. Some people call them Red Queen hypotheses in honor of Alice in Wonderland, because the virus changes, the host changes, the virus changes, the host changes, and no one ever really gets anywhere, but they both survive. Now, this all begins with Charles Darwin, of course, who formulated some of the earliest views on evolution being a process of natural selection. And this is really cool. This is one of his notebooks where he drew a phylogenetic tree. He thought organisms had common ancestors and they could be related in these sorts of trees. And back then, the way you would make these trees is by looking at, say, birds and checking out how their beaks were different and seeing who was ancestral to who. Now we can sequence the genomes of the birds and put phylogenetic trees the same way. I once saw someone in a bar. She had this tattooed on her shoulder, right here. It was amazing. I looked at it and I said, is that what I think it is? She said, yes, it's Charles Darwin's first phylogenetic tree. A modern view of what Darwin proposed is that we have an organism and there are lots of proteins in the organism. Let's just think of one of the proteins. It's been adapted over years to its environment. It works really well. Suddenly the environment changes. You go from blue to red. The host protein is now maladapted. It doesn't work well in this new environment. So we select for proteins that are now better adapted. That's called adaptive evolution. So the protein now works better in the new environment. That gene encoding that protein is then moves throughout all the organisms in that particular group, whatever we're looking at, whether it be a bacterium or a squirrel. That's called purifying selection. So all the maladapted genes get moved away. And then we have now this new protein in a new environment, which will be maintained until the environment changes as well. So this is basically the molecular view of what Darwin figured out. Now, if he were around today, he'd go crazy with viruses. He would absolutely love them because they are the best exemplars of evolution by selection, exactly what he proposed. You can see that in viruses more than in any other organism. And in fact, if you study an RNA virus, evolution is so quick, you can follow it in real time. You can do it in a day in your laboratory and watch evolution. So he would just absolutely love viruses. I would predict he would study them, and he'd probably go to some island somewhere to study them as well, because I understand he liked to do that sort of thing. So evolution of viruses is redefined as the constant change of a population in the face of selection pressures, really not very different from what Darwin originally proposed. And we'll talk a little bit today about these two questions, where viruses came from. We'll talk about that at the end. Unfortunately, I could do a whole course on evolution. It would be really interesting. Maybe someday I will. But for now, we'll spend a little time talking about where viruses came from. We don't have a lot of time to do that. But where they're going, well, I want to talk about the mechanics of evolution and how that works. As I've said already, in talking about Charles Darwin, virology today provides a wonderful window into how evolution works. And not just viruses, but the ability to sequence genomes, as said, has really revolutionized this field. So, for example, host populations grow. Viruses emerge that can infect them. And because of this, new populations emerge every day. And we see this happening with the ability to isolate and sequence viruses. But it's also clear that viruses select host populations. We see many examples where host populations are rapidly changed by the introduction of a new virus. Next time when we talk about emergence, we'll talk about how the introduction of viruses from Europe to the New World wiped out populations of people who were living here. They shaped them, and they continue to do so today. Essentially, if you cannot adapt to a new virus, you're going to be eliminated. Now today, medicine, modern medicine, in many ways bypasses natural selection. We can give you medicines, we can give you vaccines that will allow you to survive where maybe you would be otherwise wiped out. It's a very interesting theory. In fact, the dean of Columbia Medical School just wrote a book all about this. He looks at properties of humans today, and he suggests that many of them wouldn't be here if it weren't for modern medicine. Jim Goldman is his name, in case you want to check it out. The public sees viral evolution every day, okay? And you know, a lot of the public doesn't believe in evolution. I get letters all the time from people saying, you say this virus is 100 million years old, but I have to make a leap of faith to believe that. And people don't believe in evolution. But the fact is that new viral diseases are always emerging. We see them every year. We got AIDS, West Nile virus in the US, hepatitis C, Ebola virus. This year it's Zika virus. Next year it'll be something else. We get influenza and common colds every year. We get drug-resistant viruses. This is all evolution. Viruses evolve faster than many people can really understand. And even if you don't believe in evolution, it's staring at you right in the face. You get a virus infection, that's evolution. Now, as you'll see in a moment, the science writers out there don't understand this either. And I, every year, hope to release a few science writers into the wild, hoping that you will get it right. All right, first of all, 4 very important drivers of virus evolution. Anybody going to be a science writer one day? Okay. Do not forget what you learn here, okay? Promise? Because if I catch you writing things that are wrong, I'm going to write to you. I'm sure you will remember it. All right, there are 4 main drivers of virus evolution. Got to remember these. This will be, in some way, on the final exam. Large numbers of progeny. So right away you can realize that humans don't evolve very quickly because we don't make large numbers of progeny. It takes thousands of years to see an adaptive change in humans, but not in viruses, especially if the generation time is 20 minutes. Large numbers of mutants. We make a lot of mutant cells, but they don't get transferred to our germline. Quasispecies effect. This may be something you don't understand. We'll talk about that. Maybe you've never heard it before. And of course, selection. Selection acts on what is made by mutation, and that's how evolution occurs. So let's examine how this happens. We've already alluded to in this course the fact that viruses make lots of progeny when they infect cells. So here are 2 examples just to remind you. Hepatitis B virus. The virus turns over every 24 hours. The half-life is 24 hours, 50% turnover daily, total production of blood daily, over 10 to the 11th virus particles. It's a huge amount of virus. HIV, the half-life is even shorter, 6 hours. The daily turnover, this is remarkable. 90% of all the HIV made in one day, about over 10 to the 9th particles, turns over in 6 hours. So when you make new genomes, you have new mutations all the time. And this is the key for viruses. They replicate quickly. Every time a genome replicates, you get mutations. And if there is some selection to work on that mutation, you will select a new genome. Now remember, we don't always know what the selection is. And this is why I always warn you not to anthropomorphize when it comes to viruses. You may think, well, this virus was selected because the climate is warm and these mutations stabilize the virus against heat. How do you know that was the selection? You're thinking, yeah, it's hot and it would be nice to get around the heat. But maybe it's something that doesn't make any sense to humans at all. So the selective pressures are often unknown. We alluded last time to the fact that whenever a genome replicates, errors are made because polymerases are not precise. And evolution depends on mutation. That's the only way that evolution occurs. If you take away mutation, you will get no evolution. Life as we know it on the planet, every organism on this planet undergoes mutation every time nucleic acid is copied. If you take mutation away, there'll be no more life. It would not be able to adapt to changing environments. So mutations are made during copying of a genome. In fact, viruses are always mutating. And what bothers me more than anything is when a headline appears saying the virus is mutating. Here, the Ebola virus is mutating in Time magazine. Please do not ever write a headline saying Ebola, Zika, Influenza, HIV is mutating, because every minute of every day, every virus is mutating. What's the point of telling us this? What they have done is misinterpret what the scientists have told them. Scientists at a French research institute say Ebola is mutated, and they are studying whether it may have been more contagious. They're studying whether it has a phenotype that's different as a consequence of this mutation. Now that's better, but somehow it didn't make it to the headline, and you will see this over and over again. Just search "the virus is mutating" on Google, and you'll see how many articles you find, and they're all absurd in my view because they don't tell you anything. This is one of my pet peeves. If you hadn't noticed, I have some. All right, RNA viruses, last time we talked about these, make an error of about 1 misincorporation per 1,000 or 10,000 nucleotides polymerized. So in a 10 kb genome, you get 1 mutation per genome. Every time the genome replicates, you get at least 1 mutation in a 10 kb genome. So that's quite a bit. We talked about this being a consequence. All polymerases make mistakes. Okay? DNA and RNA polymerases all make mistakes. But the RNA polymerases have no error correction that we talked about last time, as the DNA viruses do. So DNA viruses are not as error prone. They make mistakes when the DNA polymerase copies the nucleic acid, but they can correct it. As a consequence, their error rate is far lower, and they generate less diversity than RNA viruses. So here's a very nice graph where we're looking at, on the x-axis, genome size from very small, 100, and there are some virus-like elements, we call them viroids, with genomes of 100 nucleotides. We'll talk about those later, and they encode no proteins. They have a very high mutation rate. So on the x-axis, genome size from 100 to 10 to the 7th nucleotides, and then on the y-axis, mutation rate in substitutions per nucleotide per generation, from -10 all the way up to -2. So you can see bacterial cells, which have, of course, double-stranded DNA, they have very good error correction mechanisms. They have a low mutation rate. And our viroid up here, which is single-stranded RNA, has the highest mutation rate. And then you can see some RNA viruses in yellow and white and red. They're all clustered up here with a relatively high mutations rate. Our DNA viruses, let's see, single-stranded and double-stranded DNA. You can see double-stranded DNA is quite low, about 10 times less than the RNA viruses. So this shows you the error rate of these viruses. And again, the ones on the upper left have a higher mutation frequency, and they're likely to evolve much more quickly. We talked about large numbers of progeny, high mutation rates. We're going to put them together in a moment. But first, quasi-species concept. What is that? So this derives from a paper published in 1978. It was called Analysis of an RNA Bacteriophage Population, Q beta. And this was just about before sequencing, but they were able to look at the genome in a way that hadn't been possible before. And let me read this to you. The Q beta, which was a bacteriophage population, is in a dynamic equilibrium with viral mutants arising at a high rate on the one hand, and being strongly selected against on the other. The genome cannot be described as a defined unique structure, but rather as a weighted average of a large number of different individual sequences. So he called this, the authors of this paper called it a quasi-species, which is not really, it's turned out not to be the best description, because species, we know today that species are different in their genomes. For example, every cell within us is different in its sequence from every other cell, but we're still a species. And species really has no meaning for viruses, but the name has stuck. People who study these phenomena nowadays call it something else like swarms and so forth. But we'll use quasi-species because that was the original description. Someday we'll change it. Now, this wasn't appreciated by most virologists, including myself. I had no idea what this paper meant. This was very early in my career. At this time, I was doing my PhD, and I didn't know what a quasi-species was or what implications it had. But I learned over the years, and I'm going to impart to you some of that knowledge. Basically, what this means is that virus populations are not one sequence. When I show you a sequence of a virus, and I'm going to show you one in a moment, that's not what the virus genome is like. The actual genome is a mixture of different sequences, non-identical but related. We call this a quasi-species. So that is what a quasi-species is. So here is a visualization of it. On the right is the sequence of poliovirus RNA. I determined this as a postdoc between 1979 and 1981. It was one of the first viral genome sequences to be determined. I was very excited about this. But guess what? It has very little to do with what the genome actually looks like, because the genome of any virus is a quasi-species, which is really what's shown on the left. So each of these lines is a single genome. You could say it was polio, one of these here on the left, except the differences among the genomes are shown as little stars and triangles and squares and so forth. You can see that almost every genome differs from all the others. That's what's meant by a quasi-species. And now that we can do very, very deep sequencing of RNA genomes, we know this is true. We don't know the actual extent of the quasi-species. For example, is every RNA in a tube of polio different, or is a certain fraction different, and so forth? We're getting to that answer because sequencing methods are associated with errors, enough errors to confound understanding what a quasi-species exactly is. But it really looks like something on the left. The details are what we need to figure out. All right, so a genome is a quasi-species. It's not a single sequence. So you may ask, what is it that you determined? I determined the average of all the different genomes. So that's what that is on the right. And it may be that there isn't a virus with that sequence in the entire population. So when you initiate an infection, whether you're infecting a cell culture or an animal, you're not using one virus with one genome. You're using a quasi-species. It's a mixture of different sequences. And they all go into the host. And selection, very likely, results in the ones that are different coming out. Maybe they have a particular advantage in the host or in cell culture. But there are selective forces that will modify that quasi-species. And then when they go from host to host, they will be further modified. And people have done very interesting experiments where they have restricted the quasi-species of a virus experimentally, and they show it doesn't do well when you apply pressure to it. So a quasi-species is absolutely necessary for viruses to be able to infect efficiently, presumably because in every cell they infect, there's some kind of selection that's going on in it. In a dish of cells, you may think every one of those cells looks identical, but they're probably not. And the same thing goes for hosts. There are different kinds of selection. So if you remove the quasi-species, you remove the ability of viruses to effectively replicate. I want you to get used to this idea of the myth of consensus genome sequences. If you go to the PubMed website, you can search for any virus and find a genome sequence. All right? And that has been determined using techniques that I used years ago, and you get a consensus. And so they will report, you look up Zika virus from Brazil on a certain day, you will get a single sequence. You will not get a quasi-species. First of all, because you would have to have thousands and thousands of entries. And what you get is the consensus, which is what I got back in the 1970s. So for every virus population, the genomes cluster around a consensus or an average sequence. And we don't know if that sequence is actually in the population. So that is really key to understand. It's a little confusing, but again, next time you look at a sequence, you'll look at it differently. That's not exactly what's out there. Mutation generates a quasi-species. The mutation of the polymerases that we've talked about, DNA or RNA, that's the main maintainer of a quasi-species, but also there are two other important factors, recombination and reassortment. So when two cells are infected with two different viruses, we talked about this a long time ago, the polymerase may exchange templates. If there are two different viruses, a blue and a green virus, the polymerase may exchange during copying of one, and you get a hybrid molecule. That's a recombinant molecule. This happens for some RNA viruses, and it contributes to the quasi-species diversity. And reassortment, viruses with segmented genomes, when two different viruses infect the cell, you have mixtures of the segments arising. You get reassortance, and that contributes to the quasi-species as well. So mutation, recombination, and reassortment are all part of generating the diversity. That diversity, that quasi-species, which exists for both RNA and DNA viruses, by the way, it's just of a lower magnitude for DNA viruses. That diversity is the raw material that selection acts upon. And whatever the selection might be, a new host cell or a new host animal, or subtle things that we don't even know about. You know, we know about the obvious selections, but we don't know about most of them that are going on inside every infected cell. So in this case, Survival of the fittest would mean a rare genome with a particular mutation would survive a selection event, whatever it may be. And that mutation then is in all the subsequent genomes. If a virus goes into a new cell, which would be an extreme example, but it's one we can readily think of, a single amino acid change would be selected for because it allows better replication in that new cell, and that change would then be found in all the progeny virus. Viruses that don't have it aren't going to be represented. But there, you can imagine, and I think this makes sense, a lot of other mutations in the genome that aren't positively selected for by this selection event are going to come along for the ride as well. We call that survival of the survivors. The linked but unselected mutations come along and get a free ride. And those may have a role later on in another selective force that occurs. So this is why having a diverse population, a quasi-species, is so important. It gives the virus flexibility to be able to adapt to new environments. And again, I'm saying this over and over. I think the selective forces we have no clue about, and they are multiple. They happen within a plate of cells for sure, from cell to cell. They happen from different cells to cells. They happen depending on what you add to the medium. And of course, in animals, they're even more diverse as well. We've only identified a handful of selection pressures because it's very hard to do so. But there's certainly many more that act to modify these virus populations. You may conclude that, in fact, this whole process of diversity is a positively selected attribute in itself, right? So it sounds a little funny. We're talking about making mutations so that you can overcome a host switch or a cell switch. But the actual process of generating diversity, the process by which polymerases make mistakes, is a phenotype. It's a property that's actually selected in itself. And in many different systems, mutations have been introduced in viral polymerases that reduce the frequency of incorporation errors. There's one very well-known one in the poliovirus RNA polymerase, and that molecule is shown at the upper right. We talked about RNA-dependent RNA polymerases a long time ago. Remember, the palm domain of these polymerases is the active site, where there's a mutation at glycine 64 up there in green. If you introduce this mutation into the gene encoding the RNA polymerase of polio, it will make the enzyme make fewer errors. It will improve its fidelity. So this was an artificially selected mutation. You don't see this in nature, and that may be telling us something right away. But if you take a virus with that change and you mix it together with wild-type virus, that virus doesn't outgrow the wild-type virus at all. And what we conclude is lower rates are neither advantageous nor selected for in nature. Many experiments have been done in competition style, which show that making fewer mutations doesn't help you at all. In fact, it hurts in many situations. If you infect an animal with two viruses, a wild-type virus and another one that makes fewer mutations, the one that makes fewer mutations always loses. It doesn't cause disease. They lose pathogenicity. So when you go from a cell culture to a complex infection in an animal, the ability to make mutations is absolutely essential. So high mutation rates are actually selected for during virus evolution. This has also been confirmed in bacteria and archaea as well. If you make polymerases that make fewer errors, the organisms do not do well. So surprisingly, or maybe not surprisingly, this is a property that has been selected for in evolution, making mutations. And that shouldn't be surprising in the end, because mutations are the basis for natural selection, of course. Talking about mutation frequencies, you may think that, oh, this is great. Let's make as many mutations as we can because it will give the virus the ability to be introduced into new environments. Well, in fact, that's not entirely true. There is what we call an error threshold, all right? And this error threshold balances mutation with survival. In other words, if you go beyond this error threshold, the error threshold is a level of mutation, a number of mutations, say, per replication cycle. If you go over it, if you make too many mutations, you will lose infectivity. Conversely, if you go far below the error threshold, you don't make enough mutations to survive, you can't survive selection. Now, this error threshold, and I'm going to show it to you experimentally in a moment, because right now it's a little bit obscure. RNA viruses evolve very close to it. So if the error threshold is where my hand is, RNA viruses are just below the error threshold. It's very easy to push them over it and you lose infectivity. DNA viruses are way below the error threshold. And largely that's because they can't make that many mutations. Their polymerases have error correction machineries. All right, so even though mutation is great, you can't make too many mutants and you can't make too few mutants. You have to be at a certain place. So here's an experiment where we can demonstrate the existence of an error threshold, first for a DNA, then for an RNA virus. So we take a DNA virus and we infect cells with this DNA virus, and we have added to those cells a base analog. So 5-azacitidine is a base analog. And what happens is this will be incorporated into the DNA, the replicating DNA of the virus, sort of like an antiviral chain terminator. But in this case, it gets incorporated as a C, but behaves as a T when the polymerase copies it. So you get, instead of a G, even though it's 5-aza-C, the polymerase, when it moves over it, it will put in not a G, but it will put in an A. It looks like a T to the polymerase. So it's introducing lots of mutations into the genome, and that is why this drug is a mutator. So the mutation rate among the progeny of an infection where you've added this drug to the culture increases several orders of magnitude. 10, 100 times more mutants are made by adding this 5-aza-C. Now, if you do a similar experiment with an RNA virus, you use, again, a base analog that increases the mutation frequency, you only get a 2 to 3-fold increase in the number of mutations in the progeny viruses. So a DNA virus, you can increase the mutation frequency several logs, 10 or 100-fold. The RNA virus, only 2 or 3, and that is because the RNA virus is existing close to the error threshold, this point at which you can't exceed, otherwise you lose infectivity, and the DNA viruses are existing way below it. All right, so that is the error threshold experimentally shown. So here's a graph of one of these experiments. This is with an RNA virus. And what we are doing here is we're looking at the infectivity of the genome on the y-axis. So we start with wild-type virus untreated, which is 100%, we set as 100% infectivity. And then what we are doing is we are doing infections in the presence of a drug called ribavirin. And ribavirin is very much like 5-aza-C for RNA viruses. It causes mutations to be introduced into the viral genome. So we add increasing concentrations of ribavirin, and the result, you can then take the viruses from the experiment, you can sequence their genomes and ask how many mutations have you introduced into the viral RNA. So here are the mutations per RNA genome on the x-axis. We go from 0 in wild-type virus to 16 or so. And you can see, if you introduce just 2 mutations into the genome, you already lost 80% of your infectivity. So that's the error threshold. You put 2 or 3 mutations in, 6, you've lost most of your infectivity. So it shows you that you can't, an RNA virus is already existing very close to the error threshold. You can only increase the mutation frequency 2 or 3 times, and then you exceed the threshold and you lose your infectivity. So the point of all this is simply to show you that there is a range where viruses exist in terms of the number of mutations they sustain per genome. It is highly regulated. It's just not occurring willy-nilly. There's a certain threshold and the viruses have to be at or below it. Now, another concept that helps us to understand virus evolution is the genetic bottleneck, all right? So we define this as selective pressures. If you put selection pressures on small populations of viruses, and this would be a very small number of virus particles, so you don't have a lot of genetic diversity. When you put selection pressure on that, the virus doesn't do well because there aren't enough variants to emerge from the selection process. So, and we do this, we illustrate this with a very nice experiment where we take an RNA virus and we do a plaque assay, shown on the lower left. You can see all the nice plaques, and you can pick one of those plaques. So it's a little jargon to say pick a plaque, but you're taking a pipette and removing the agar over the plaque, so you're basically removing the virus from a single plaque. You then take that virus and infect another plate. You do a plaque assay, and then you pick a plaque, and you do it over and over and over. You just keep repeating this from plate to plate to plate. So it's plaque to plaque passage. Now, one thing you have to remember is in a single plaque, there are not a lot of viruses, maybe 10,000 or so. So the diversity is highly restricted, and that's the key to this experiment. For many viruses, after about 20 to 30 of these plaque-to-plaque passages, you get a really sick virus population. The viruses are barely able to grow. They're not fit. They don't grow well. They don't do well when you put them on new cells. So what's going on here? You're using the same cells. You're using the same medium. The only selection is this plaque-to-plaque passage. I remember this happened years ago. I had a student in my lab who was trying to amplify a virus she had recovered, and she was going plaque to plaque, and it would never grow beyond a certain point. And I said to her, don't you remember virology course, the genetic bottleneck that I'm telling you about here? And that's in fact what explains this. So why is the fitness of this virus so low? The reason is a genetic bottleneck in which the diversity is restricting the ability of the virus to adapt to a new environment. And the bottleneck here is a few thousand viruses derived from a single virus. So here's an illustration of a bottleneck. You have a big bottle of different viruses, but if you try and squeeze some through the bottleneck, only a few come out. And the bottleneck here is the plaque assay, because you're just picking a single plaque. So you have a few thousand individuals, and those are expanded, but their genetic diversity is very restricted. If you could expand the whole bottle's worth of viruses, you'd be way better off. But even just doing a little growth in culture with such a limited population is a problem. So again, after 20 to 30 passages, the virus loses its ability simply to grow to good titers in cell culture. Now, this phenomenon of passaging virus with low population diversity and having a defect in virus replication, this is called Muller's ratchet. And it was originally found in other organisms. Small asexual populations decline in fitness over time if the mutation rate is high. And replicating RNA viruses, of course, are close to the error threshold, so they fit this bill. And what we have done is we have restricted the population of this RNA virus to single founders in the plaque. Remember, a plaque is initiated with a single virus. So you're really putting a severe bottleneck on the population by selecting for the offspring of a single virus. And many mutations basically accumulate in these viruses. The virus exceeds the error threshold, and you lose infectivity. We know this now because we can sequence these populations and sort this out. So the implication is that if we had more diversity, we would overcome this bottleneck, because we would have a whole range of mutations to select for, not just the limited number that are found in these serial passed viruses. So this is called Muller's ratchet. It's a metaphor which is used to describe how the mutations work. So you know, a ratchet is this gear that can move only in one direction because it has a ratchet which engages the teeth of the gear. And that's what's happening here. At each passage, these viruses are accumulating mutations, but you can't remove them from the genome. So the mutations accumulate, but they never go away. And after so many passages, you've accumulated so many mutations that the virus is no longer infectious, clicking relentlessly as mutations accumulate over every cycle. I'm going to explain that to you in a minute in terms of how we can fix it. But let's look at it diagrammatically. Here we are back to our quasi-species illustration. So here's our initial population of viruses. If you grow the virus, say in liquid culture, where many cells can be infected and the population can amplify to high titers, you will have diversification of that initial genome and good replicative fitness, because you'll have high diversity. Now, if you do a plaque purification, you then amplify it, and you get the same thing really repeated, and you can see some of these mutations are present in all of the viruses. And if you go plaque to plaque, you're just going to reinforce this. On the left is growing the virus in liquid, where we get good amplification of all the genomes. On the right, if we plaque purify that single virus, you then have a number of mutations that are always present, and the diversity is clearly restricted. You can see that from the picture, and that gives you the decreased replicative fitness. Does this really occur in nature? Yes, the answer is yes. And here are some more examples. These are 5 different viruses: a bacteriophage, and two bacteriophages and 3 animal viruses. And in the second column, the number of passages that were done on these viruses. Again, plaque-to-plaque or very low dilution passages. And you can see the % decrease in fitness that accompanies this passage. So it varies for each virus, but look at HIV. After only 15 bottleneck passages, you have a 94% decrease in fitness. And so these viruses, I think the lower passage and higher effect means that these viruses depend on genetic diversity to be able to survive in hosts. And in fact, these are just in cell culture, these passages. So immediately you should be thinking when a virus is going from host to host, you have to somehow get around bottlenecks. Otherwise, the virus is not going to be successful. Now, in the real world, are there bottlenecks? And absolutely, there are. So you look for situations where infections from one host to another occur with very small inocula of viruses. So, for example, the small droplets of virus that go from one person to another during aerosol transmission. You know, when I sneeze or cough and I send out a droplet containing viruses, there are only a few thousand viruses in that droplet. So it's like a bottleneck in a way. And that's going to grow in a person. That could go to another person as a small droplet and so on. Activation of a provirus or a latent virus from a limited number of cells. Activation of herpes simplex virus from a peripheral ganglia. It's a single genome which is being amplified, similar situation. Or when you are bitten by an insect, you don't get a lot of virus by that. So there are lots of examples of this. So you may be asking, well, okay, there are situations in nature where you get a little bit of inoculum. How do we get around that? Because obviously viruses do well in nature. And so here's the answer. How do you avoid the ratchet? Nurse ratchet I'm thinking about, right? Well, don't pick a single plaque. If you are going to amplify a virus, pick several and mix them together. So in the laboratory, that would help you a lot. Essentially, any way that you get more diversity in the population, that will help you make a mutation-free genome. And how does that happen? Well, the main drivers of making a mutation-free genome are recombination or reassortment. They remove the mutations that are deleterious. Because if you have a big enough population, even if you have lots of mutants, they could recombine or reassort and make a virus that's now fit. So let's take a look at how this happens. So we've done a limited diversity passage, and we've made a couple of sick viruses. Sick virus 1 has a mutation here in the purple, and sick virus 2 has a mutation somewhere else. So each of these don't replicate well. Now, if they're both present in the same stock of virus, they could undergo recombination to get rid of the deleterious mutations and give you a wild-type virus. So that process depends on having enough genomes present so that when a cell is infected, it could be infected with 2 different genomes like these, and you could recover the loss of infectivity. So recombination and reassortment, which I haven't shown here, the swapping of segments. These are really important for restoring diversity in situations like when you are bitten by a mosquito or you acquire infection by a respiratory aerosol where there are few virus particles present. This is actually a very nice summary to this whole discussion about the quasi-species. Diversity of a population is important for survival. So we can remove diversity by making the polymerase more accurate. Population survival plummets. We can also remove diversity by restricting the passage to small populations that accumulate lots of mutations but don't have a wide variety of mutations. Population suffers as well. So 2 ways of illustrating this really important driver of mutation. Let's talk a little bit about how these are selected upon. And I want to give you an example of 2 that we call genetic shift and genetic drift. And we have talked about this briefly before, but let's define it again. When you have an antibody response to a virus, when you have an immune response to a virus, whether it be antibodies or cytotoxic T cells, these apply a pressure on the virus population. And if there are enough mutants in the population, they could be selected for, say, mutants that escape antibody neutralization or mutants that escape CTL killing of infected cells. Because remember, in both cases, a viral sequence is being recognized. An antibody binds to a virus particle, or a CTL recognizes a virus peptide presented in a MHC molecule. This is a very nice example of one selection force that we're very aware of, the adaptive antibody, the adaptive immune response in immunocompetent individuals, of course. This results in two kinds of diversity. You get drift, diversity arising from copying errors. So every time a genome is replicated, it incorporates errors. And if one of those errors happens to be in the antibody combining site on the virus particle, then those viruses could arise. They could be selected for because they don't grow in you in the presence of your antibody. And another example, more drastic, is what we call genetic shift. And this is diversity arising after either recombination or reassortment. So it's not mutation caused by error-prone replication, but it's exchange of sequences from a different virus, and that also will allow you to escape the antibody response. So let's take a look at how this works. And of course, the master of these two ways of escaping, I should say the master of selection by these two different ways, are influenza viruses, which we've talked about very often previously. These are envelope viruses with two different glycoproteins in the envelope, HA and NA. And in fact, we classify influenza viruses by the serological nature of the HA or the NA. And we typically call influenza viruses H, X, and Y to designate the origin of the hemagglutinin and the neuraminidase. And so far, we have identified 18 different hemagglutinins out there and 11 different neuraminidases. And all of them, with one exception, the number 18 is a recent isolate from bats. All the other 17 infect birds. And only one, H1, H2, and H3 can infect humans and be transmitted effectively among them. So all the strains of influenza virus we see every year globally in people are either H1, H2, or H3. Right now we have H1s and H3s co-circulating. We don't have any H2s around as far as we can tell. Now these are all rising by genetic variation. So here's an example of genetic shift. Back in 1918, there was a pandemic of so-called Spanish flu pandemic, where many millions of people worldwide were infected by a virus that the population hadn't seen before. There was no immunity to this virus. It appears to have originated from birds. And we were able to reconstruct this virus recently, as I told you some time ago, but we don't have the bird origin of that virus, so we don't actually know what the bird virus looked like, but we can tell that most of the segments from this virus look avian. So the idea is that this virus jumped from a bird into a human and caused very serious disease. That virus stayed in people for many years until 1957, when all of a sudden a brand new virus arose called H2N2 virus. And this virus was antigenically brand new. Both the hemagglutinin and the neuraminidase had not been seen before by people. And so this virus caused a pandemic, spread globally, caused a lot of infection. Now that virus is a reassortant. It is a reassortant, and you can see the red genes come, or the red RNA segments come from the 1918 Spanish influenza virus. And the yellow segments came from a H2N2 virus that was circulating in ducks in the early 1950s. What is replaced are the segments encoding the hemagglutinin and neuraminidase, as well as a few others. One other, the PB1, one of the polymerase genes. So you can see we have a brand new HA and an NA coming from a duck virus. It had never been seen by people before. This virus causes another global pandemic because we don't have antibodies to it. An example of genetic shift. The mechanism is reassortant. Now in 1968, another pandemic emerged, the H3N2 virus. And this virus acquired the HA from a duck influenza virus, and that H3 came from the duck virus. You can see it in blue there. The N2 was maintained from the H2N2 virus. And again, the H3 was a brand new virus for humans, so we had another global pandemic. This happens roughly every 20, 30, 40 years. It's not terribly regular, but it happens, and we see these three occurrences all in the 1900s. The most recent pandemic of influenza virus was in 2009, when the 2009 H1N1 strain emerged, and most people in the world were not immune to this virus, so it caused a global pandemic. They call this swine flu because it seems to have originated in pigs. And this is also a reassortant. And it's a very complicated reassortment. Look at all the viruses that gave rise to this. You could look at the colors of the RNA segments, and they come from a variety of viruses. Now, that 1918 influenza virus, when it infected humans, it also infected pigs. And the pig population of the world has been steadily increasing because pig is a great source of protein, and many countries raise many, many millions of pigs every year. And it turns out they're really good hosts for influenza virus. So that 1918 virus went into pigs, which have since been shipped all over the world. So we actually have a lineage of influenza virus that infects Eurasian pigs, and we also have a lineage of H1N1 virus that we call the classic swine H1N1, which infects pigs in other places as well. This 2009 virus was a reassortment of the Eurasian swine influenza virus, a classic swine influenza virus, a human H3N2 virus, which had been circulating since 1968, and an avian influenza virus as well. And there were some intermediate reassortments that occurred, but then this one, the final one that took off, it contains genes from all these viruses. And this virus was initially seen in Mexico, and it's thought that it might have emerged on a pig farm there, but we really don't know exactly where it emerged. But you can see that these viruses come from influenza viruses that infect humans, birds, and pigs. That's where all these viruses are residing. And periodically, we're going to get new ones because the number of influenza viruses in birds, 17 different HAs, 11 different NAs, there's a lot of variability. So we're always going to have new strains arising by shift unless we make a universal flu vaccine that can get them all before they infect us. We're talking about raw material for selection. So the formation of reassortments gives you raw material for selection for drift. The formation of point mutants by error-prone replication is also the basis for selection, and that can lead to antigenic drift. And we've mentioned this before. This is an HA molecule from influenza virus, and at the top, in the different colors, are epitopes where antibodies that block infection will bind. And so if you're vaccinated against influenza virus, you'll make antibodies against some of these epitopes. It only takes a 1 amino acid change in any of these epitopes to make the antibody less effective at neutralizing, and then you get a yearly outbreak. So this is antigenic drift, and it typically happens on a yearly basis. And again, the reason it happens is we have error-prone RNA replication, and among the errors, there's some changes in the coding region of the HA shown here. And if you have a person who's immunized, those antibodies will bind all the viruses that don't have the mutation, but maybe there's one virus with a single amino acid change, and that person will then replicate and take over, and that person will transmit it, and then that 1 amino acid is fixed in the population, and the next year the vaccine doesn't work. That's why we need to do surveillance to identify these issues. Now, I've been talking so far about viruses mutating and recombining and reassorting, but that's only half of the picture, right? Because there is a host, and hosts mutate as well, although far more slowly than do viruses. And only recently have we been able to see this, to see that viruses and hosts mutate in turn. Well, let me show you, this is a whole new field of study where we call it the host virus arms race. And I also mentioned the Red Queen before because she's the one who said, around here we do all the running just to stay in place. And that's really what's happening here. The virus changes, the host changes, the virus changes as well. So for example, we have a virus protein which is bound by the host. This could be an antibody, or it could be an innate protein or an intrinsic protein. It doesn't really matter. It's a host defense protein. And here the host is winning because the host binds to the virus very well. So this could be a virus or a virus protein. Eventually, selection will lead to the emergence of a virus with a little bump there that can't be blocked by the host anymore. And the virus turns out to win. That's by selection using the processes we've just talked about. But eventually, new hosts will emerge. It will take a long time in the case of people, of course, but other animals reproduce more quickly. Eventually, a new host will emerge, and the host will win. It will adapt to the virus with the change, and it will be able to neutralize virus infectivity. So we can actually see this happening now. We can go back and we're sequencing every organism on Earth, and we can see this happening back and forth between virus and host. So let me show you an example of that. Let's say you make a phylogenetic tree of different primates. You can have at the root the ancestor of all primates. And you can have all sorts of primates, not just chimps and humans and apes, but all sorts of monkeys as well, arranged according to their evolutionary relatedness. And then you can look at specific proteins in these animals. So we can do this now. We can sequence the entire genome of anything that's around, and we can deduce all of the coding sequences, and we can figure out, we can align, say, orthologs, which are related proteins in the different animals. So if you wanted to look at an intrinsic defense protein, like a tetherin, you could line all the tetherin sequences of these animals. Then you can ask, are any of these lineages, or in any of these lineages, is a specific sequence of a protein positively selected? In other words, did it arise here in the ancestor and become selected through evolution? That's called positive selection. And you can identify those codons. So here, for example, you have a position in one of these defense proteins where you can see there's a selection for a specific amino acid. Then you can actually do experiments to test whether these changes make any difference. You can infect cells from these primates in culture, and you can see if the virus works. We call that virus winning, or if the host wins. And you can go along and make a tree of all. You can get cells from each of these animals on this phylogenetic tree. You know the sequence of the virus, the sequence of the defensive gene, and you can ask in which combination who wins. You can take it even further. You can then map the positively selected changes. Here we're looking at the sequence of a host gene, and we can change this amino acid in the host protein and ask who wins when we change it. Does the host win, or does the virus win? We can end up saying when the virus is winning, this amino acid is a serine. When it changed to a glycine, the host wins. We can do amazing mapping of both viruses and host defense proteins. And when you do this, you learn that these virus-host conflicts, that is, virus changes, host changes, virus changes, and so forth, these conflicts have driven the evolution of the immune system. We have a handful of examples of where this is very clear. So on the left, TRIM5 alpha, a host defense protein involved in intrinsic defense. We can see positively selected residues that have changed over years in many different primate species under pressure from immunodeficiency viruses, simian immunodeficiency like viruses. And on the right, PKR. Anybody remember PKR? That protein in the cell that responds to double-stranded RNA and then phosphorylates the IF2 alpha. It shuts down host translation. Look at all the positively selected residues. They're red triangles by different viruses. And we can map them one-on-one because we can take the virus and the PKR and make the changes in both and show what's going on. Look at all the viruses that have applied pressure to PKR. And that's because this is an incredibly important defense protein for many different viruses. But the key here, and the amazing thing, is that we can go back in the phylogeny that we have and map all of these conflicts over time. You should be getting a sense now that we have a lot of interesting genome diversity out there, but still, for some viruses, it's limited. 3 serotypes of polio, lots of serotypes of rhino, but 1 measles serotype, continuous influenza variation. Why do we have these differences? Why does flu vary so much and measles does not? Measles and flu are both envelope viruses. Why do we have this big difference? I don't have an answer for you. People are really interested in understanding. It's probably, many people think, well, there must be structural constraints of the measles glycoprotein versus the influenza glycoprotein, and that's probably part of it, but there's got to be something more as well, and it may involve the host. But I want to leave you with this question because it's really important. All right, the next topic I want to talk about is the selection for virulence. Is virulence a positive or negative trait? And the idea here is if a virus is too virulent, it will kill the host before it can transmit it, right? And maybe if it's not virulent enough, it doesn't transmit well from host to host. So if this were true, you would expect all viruses to evolve to be really infectious but avirulent. But this is not what we see, of course. We have lots of viruses that are avirulent, but we also have viruses that cause a lot of disease. And the great example I like is poliovirus in the gut. Poliovirus causes no gut pathology. In 99 out of 100 people, it replicates, is shed beautifully to another person. It transmits really well, no disease. You can't even tell you're infected. Only 1 out of 100 the virus accidentally gets in the CNS. There's no gut pathology. Then we have norovirus, which is also transmitted really well from one person to another, yet it gives you diarrhea and gastroenteritis and vomiting. It really causes a lot of pathology. Why do we have to have two? Why can't all viruses be avirulent and transmitted really well? Again, I don't have an answer for that, but I want to bring you through a couple of bits of information that help you think about it. An experiment was done in Australia that addresses this. A rabbit was brought into Australia in 1859 because they wanted to hunt. They didn't have rabbits that they could hunt easily. So this European rabbit was brought in, but it reproduced really extensively because it had no predators in Australia. So they said, crap, we have a problem. We have to get rid of these rabbits. They decided to use a virus to kill off the rabbits. A myxoma virus was introduced in the 1950s. Now, the natural host is the cottontail rabbit, and that's not the one they imported. They had cottontail rabbits, but they're not affected. The European rabbits, which they brought in, the infection is 90 to 99% fatal, and it's spread by mosquitoes from rabbit to rabbit. I don't remember which of these is which. One of these is a cottontail, and one of them is a European rabbit. They released this virus into nature, and in the first year, 99.8% mortality rate. But there were a couple of rabbits left that didn't die. They had mutations in their genome that made them resistant to the virus. It's always going to happen when you have enough offspring, right? Second year mortality dropped to 30%, which is still not insignificant, but it allowed the rabbits to reproduce. So this was a failure. Never worked because the rabbits ended up growing year after year, and they were never eradicated. So what happened here, of course, is the rabbits and the viruses make a lot of offspring. The virus probably, and this has been shown experimentally, evolved to kill fewer rabbits and extend the life of infected rabbits so the virus could be taken up by mosquitoes and overwinter. And the rabbits, of course, also evolved to become more resistant of the virus as well. And this is what we think should happen when a virus newly infects a population. Initially, it should be quite lethal, but eventually the population should adapt, the virus should adapt, and some kind of medium in between should be reached. So a nice experiment illustrating that. Now, what about people? Can we say anything about the evolution of virulence in people? We know lots of examples where viruses, in fact, all the viruses we have once started in animals. A great example is HIV, which we acquired from chimps. We'll talk about that later. But Lassa virus, Ebola virus, all these viruses hopped into people, and they're quite virulent. And these are all recent jumps. HIV in the 1920s, Ebola virus more recently. So those jumps are quite lethal. But if you look at viruses that have jumped from animals to people a long time ago, like measles and polio, these are less virulent. So the idea is when a virus first jumps species, it's not well adapted to the species, you have serious disease, but eventually over time, the virus and the host adapts, and then you have less disease like we have with polio or with measles. The problem is we've only been studying viruses for about 100 years, and we can't really see any of this happening. It's purely speculation. We can do animal experiments like the rabbit one that I just showed you, but we cannot see this in any infection of people that I know of. So while the theory is good, we have no evidence that viruses change. Nevertheless, if you look in the science news, you will find lots of examples of where people are saying the virus is becoming more virulent, more transmissible, more this or that, but you can never show it because obviously you can't infect people. And here are just some examples. Back in 2014, during the Ebola outbreak, Michael Osterholm wrote in the New York Times, it's going to mutate and become transmissible by airborne. This scared the hell out of everyone. It was pure nonsense. It never happened, and not likely that it would ever happen. Ebola virus, this scientist was quoted in an article about Ebola by Richard Preston. Is it changing so that it replicates more from person to person? No, it wasn't. It was not true at all. And more recently, Peter Hotez, a couple of weeks ago in the Times, said Zika's mutated, and that's why it's spreading globally. We always blame the virus. But there's no evidence that any of this is happening because we can't do the experiments. We don't have a historical record for testing this. People love this idea of viruses mutating. It's in all the movies about viruses. They're always mutating and getting more virulent and killing more people. Yet, if anything, it's going the other way. And the problem is we can't prove it one way or the other. We have no data on the effect of evolution on the virulence of human viruses. We have some for animals. There's a great story with chikungunya and mosquitoes that I told you a while ago. We have the rabbit story. There are others, but we have nothing for people. But nevertheless, the world has a obsession with viruses changing to kill more and more people. It just doesn't happen. It happens in the movies. It doesn't happen in real life. Let's end up by talking a little bit about where viruses came from. Now, for many years, the problem was we didn't have any fossils. So we couldn't dig up dinosaur bones and look for viruses. And the oldest viral stocks were not very old. 1918 influenza was the oldest for many years. And then a couple of years ago, a 30,000-year-old virus was pulled out of frozen ice in Siberia. So we don't have a fossil record. So what can we do? Well, it turns out we do have a fossil record. The sequencing of viruses tells us how old they are. And there are two ways. First of all, by what's called a molecular clock. You can relate the time scale of genome change. You can look at all the contemporary herpes virus isolates we have. You can calculate the rate of change per year. And when you do that, you can predict that herpes viruses arose 180, 220 million years ago, i.e. T-Rex had herpes sores, probably. Well, anyway, there were herpes viruses among the dinosaurs. But another more interesting way is you can look for viral sequences integrated into the genome of animals. And these we call endogenous viral elements. It's the whole field of phylogenomics. You sequence the genomes of different animals, and you can see pieces of viral DNA in them. You know that retroviruses integrate a proviral copy into the host. But it turns out that other viruses get accidentally integrated into the host. Little pieces of other virus genomes. Almost any other virus just accidentally gets stuck in, and we can use those as a marker for saying how old these viruses are. So here's an example where we're looking at 3 contemporary strains of rabbits, or species of rabbits. And 2 of them have an endogenous viral element at the same place in the chromosome, so you know that's an ancestor of those must have been infected. And we also have a species without an endogenous viral element. Again, we can do this because we can sequence whole genomes really quickly. So we know these 3 had a common ancestor here, an ancestral host. The ancestral host doesn't have an integration in the germline, so we can say that when these A and B split off from C, that's the maximum age of the virus, and then the minimum age would be when A and B split off as well. So we can put numbers on the age of the integration. And these turn out to be millions and millions of years. So here's an example of looking at integrations. Again, these are not the whole genome. They're just pieces of the virus. You can recognize it, and you simply use it to date when the infection occurred. So here are infections by 3 different single-stranded DNA viruses and a whole host of vertebrates, from fish all the way up to humans. And you can see we know exactly when the integration occurred on this timeline from 30 to 60 to 90 million years ago by looking at the speciation, by looking at the phylogenetic tree in a way like I just told you, and asking, was this integration site in this species, in this species, in this species? We can figure out when they went in. By that, we can figure out how old viruses are. But of course, we think they are much older than 60 or 90 or 100 million years or 200 million years. There are 2 theories about the origin of viruses, which I alluded to a long time ago. One is that they are pre-cellular. There was an RNA world where we had small pieces of RNA that were replicating in this world that had no cells, was just RNA. And we think there are viral genes which are colored black and gray. We think that viruses actually were part of this RNA world. And eventually, when cellular life arose, these viruses changed to be able to infect the cells rather than replicating outside in solution. So that's one theory, pre-cellular origin. The other is that viruses are actually escaped host genes. We had cells arising first, and then genes escaped to form virus particles. There are proponents of both theories. Obviously, it's very difficult to have proof of this, but the more sequence analysis we do, the more evidence we can get. The problem is, when you go back billions of years, right, to pre-cellular life, evolution has taken place that whole time, and it's very hard to recognize viral sequences anymore. So it may end up being very difficult to prove this. However, both of these ideas are supported by the discovery of protein structures that are conserved among different viruses, and they have very little sequence similarity. So a polymerase, for example, or a capsid protein, they could be very different and have originated in some of these ways. Human viruses, all the types of viruses that we have today, in which we continue to acquire, they've all been around for a long time, and they have infected our ancestors, not just Homo sapiens, but our ancestors, our hominid ancestors, and probably before that as well. So all of the viruses you have in you evolved from animal viruses. None of them were created de novo in us. So for example, smallpox virus sequence analysis suggests that smallpox arose from gerbils. Gerbils many years ago were carrying a related virus, and sequence analysis shows that it hasn't diversified much in people. So the idea is that it's a zoonotic infection, an infection we acquire from animals. Smallpox virus came initially from a gerbil and spread throughout the human population, causing enough death and destruction that we had to eradicate it a number of years ago. That's one of many examples. All the viruses we have today in us originated from animal viruses, and as we get new ones, they come from animals as well. Another example is measles virus. This is related to rinderpest virus, which is a pathogen of cows. And the thinking is that measles evolved from an old rinderpest that we got when we started to domesticate cattle. We said, oh, these animals are good, we can use their milk, we can eat their meat, so we began to domesticate them, and then we started to make cities where lots of cows were put together, and we got infected with rinderpest virus. This happened about 5,000 years ago when cities were just forming, and then they formed not in the Western Hemisphere, but they were brought there eventually by colonization and migration. And these viruses wiped out many Native American populations. We'll talk a little bit about that next time. It's an example of how you introduce, this is good evidence for this whole theory, when you introduce an animal virus into people, initially it's very virulent. The population is totally non-immune. They haven't adapted. There haven't been any selections to this virus, and measles and smallpox virus wiped out these populations. It takes a long time for selection to occur to make more resistant populations. If new viruses are only going to arise from the ones that we see now, so we don't see all the viruses that are out there, of course. Many of them are in animals that we have no clue about. But let's assume that any new viruses will come from the viruses that are out there. What are all the possible mutations in a virus genome? It's an interesting exercise. Sequence comparisons of a number of RNA viruses have shown that you could change over half of the nucleotides in the genome, and it's okay, the virus will still replicate. So there's quite a plastic genome. So for a 10 kb viral genome, you could have 4 to the 5,000 different sequences, okay? 4 different bases to the 5,000th power. And that's assuming 50% of the changes in a genome can be sustained, the number I just told you. And to that we can add deletions, recombination, reassortment, so we get even more changes. But let's say it's 4 to the 5,000. This is a huge number. These are all the different permutations of sequences that we can have. How huge is it? There are only 4 to the 135 atoms in the visible universe. That's a lot. But it's the possible virus mutations eclipse that by many orders of magnitude. There are lots of possibilities for making new viruses out there. But no matter how many mutations you could put in a genome, a herpes virus always stays a herpes virus. It doesn't change into an influenza virus or vice versa. And how is this kind of stability maintained? Polio never changes into flu. Herpes is always herpes. It may get very different. It may infect different animals. It may cause different things, but it's always going to have a capsid with an envelope and a double-stranded DNA viruses. And that is what maintains this stability. There are many aspects of a virus's structure and replication cycle that confine it so that you can't introduce mutations and make it into something totally different. Some of the constraints, for example, the genome. You can't change DNA to RNA or vice versa. You can't go from double-stranded to single. That's probably because the replication strategy is fixed. You need certain viral proteins to do certain things, and you can't change those drastically. The physical nature of the capsid is another constraint, especially icosahedral capsids. You can't get a genome much bigger than can fit inside that capsid. And of course, there are interactions with the host defense proteins. If a virus changes a protein too much, it will no longer be able to, say, antagonize PKR, and it won't work anymore. So despite 4 to the 5,000 possibilities in a single 10 kb genome, there are a limited number of possibilities because many of them would change a lot of these fundamental properties. I'm going to leave you with this interesting food for thought. You know that chimps and humans had a common ancestor, and we think it took about 8 million years for ancestors of chimps to evolve to become humans. 8 million years. If you sequence the genome of the chimp and humans, we are 98% chimp. You can get this t-shirt and boast of that. Only 2% difference. 8 million years it took to change by 2%. Now, in 5 days, the polio genome, from the time you ingest it to the time you shed it in your feces, 5 days it takes to go from mouth to feces. In those 5 days, the genome of polio will change just as much, 2%. 5 days. So imagine what a virus could do with 8 million years.